Okay, good morning everyone. Good morning Sunita. Good morning. Good morning Saima. Good morning. Okay, okay. Okay, good morning Bruna Rene. Good morning. Okay, let's continue. So in the last class, we have completely understand the cells of the immune system. After understanding the cells of the immune system, we started studying about the immunogenicity versus antigenicity. And we understand one important statement that, friends, good morning Priya, please continue. We read that, good morning Tarun, all immunogens are antigens, but I said to you that all antigens are not at all immunogens. That is the statement what we have studied. Yeah, I'm little better Tarun, thank you. Continue. Yeah, so later we have studied the two examples of the immunogens. One is called as a hapten, another one what we can call it as, friends, adjuvant. And what is the statement I have given to you, friends? When compared to the hapten, the adjuvant, see, both are a type of immunogens only, there is no doubt about it. But when compared to the hapten, the adjuvant can show its action in a prolonged way. I mean to say, adjuvant can act as a immunogen for the longer period of time, and adjuvant can show its action of immunogen for the longer period of time. So that's why we can called adjuvant can act as the better immunogen. Kindly don't get confused with that particular point. And all the students pay attention. So friends, look into this. This is the hapten. If we want to introduce the hapten in the body, what we will do, friends, we will always introduce this particular hapten with the help of a carrier. So what you have to understand here, carrier is the one which is carrying the hapten into the body. So that is what we can call it as, friends, hapten and carrier conjugate. Everyone listen to me very carefully, friends. Whenever hapten and a carrier got immunized to the rabbit, say suppose example, the combination of hapten and carrier immunized into my body, how my body starts responding towards that particular hapten. In certain cases, listen to me everyone carefully, our body start producing the antibodies only towards the hapten, which means our body start recognizing only hapten as the foreign substance. In certain cases, our body start producing the antibodies only towards the carrier. In this particular case, our body starts recognizing only the carrier as the foreign substances. But in most of the cases, friends, our body start producing the antibodies again is the combination or a conjugate of the hapten also as well as the carrier also. So our body starts recognizing hapten and carrier as the foreign substance. As a result, friends, they can show they can produce the powerful antibodies. Again is this particular conjugate. Here conjugate is nothing but combination of hapten as well as the carrier. See friends, most of the time, of course, we can introduce the hapten without carrier also, but our body can recognize hapten very easily whenever it is introduced with the help of the carrier. Kindly don't get confused with that point. And friends, listen, whether it is the antigen, everyone listen, this is really important statement, whether it is the antigen or whether it is the immunogen, right, entire antigen and entire immunogen cannot be recognized by our immune system or our antibody. because friends, antibody also did not have a large enough space to recognize the entire part of the antigen or immunogen. Everyone listen, I hope you all people remember, if you look into this is the antibody, the antibody having a particular binding site for the antigen. What do you call it, that particular binding site, friends? We will be calling that particular binding site as the paratope. Kindly do not get confused with this particular point. Okay, now to this particular paratope, does the entire part, entire antigen or entire immunogen will come and associate? Not at all. Only friends, a part of the immunogen or the part of the antigen. Now you can ask me a question, sir, which part of the antigen or which part of the immunogen will come and associated with the antibody? That is a valid question. Now everyone remember, the antigen, the part of the immunogen or part of the antigen which is immunologically active or which is biologically active. In other words, the part of immunogen or the part of antigen which can stimulate the biological activity of the antibody, that part we will be calling it as the epitope. So every student has to keep it in your mind. What is epitope, friends? Keep it in your mind. Epitope is the immunologically active part of the immunogen. Kindly do not get confused with that particular point. Now friends, that immunologically active part of the immunogen which we can call the epitope, where does the epitope will come and attaches, friends? Epitope will come and attaches to the paratope of the antibody. So the point you have to remember, antigen associated with the antibody with the help of a epitope and paratope. And we know very well, good morning Shivani, continue. And we also know very well how does the MHC molecule and T helper cell attaches, friends. This is more important. You know very well, if the antibody wants to be produced, the T helper cell has to activate it. It has to release several cytokine molecules. That cytokine molecules only will be helpful. We all people commonly know about it. Now every student just listen my words very carefully, friends. Just understand what exactly I am saying here. This is what, friends, antigen presenting cell or APC. What is that? You take an example of macrophage. Good morning everyone, please continue. We know that antigen presenting cell like macrophage or the dendritic cell, and this is what cell, friends? T cell. We know that antigen presenting cell on its surface, what it is having? It is having the major histocompatibility protein or the MHC protein. Once it process its particular antigen, every student listen, there will be a particular TCR receptor where this particular TCR receptor is expressed. The TCR receptor is expressed on the T helper cell also, even in the cytotoxic T cell also. Friends, now the TCR and class 2 MHC associated with each other, by that TCR receptor can be associated with the antigenic peptide. And friends, this is one of the major stimulatory signal. Which one? Class 2 MHC and the T cell receptor is the major stimulatory signal. And friends, then which one is the co-stimulatory signal? Every student keep it in your mind. The co-stimulatory signal is the CD4. We all people know very well. If it is the T helper cell, CD4, and if it is the cytotoxic T cell, CD8 and class 1 MHC comes into the picture. Don't get confused with that. Here, what is the major receptor? Major receptor is the TCR, T cell receptor. Either TCR alpha or TCR beta. Along with that, the most important co-stimulatory receptor, very important receptor, they are also CD4 or the CD8. So in case of a HIV virus, what is happening, friends? Our T helper cells are under the attack by the HIV virus. HIV virus exclusively attacking the CD4. When the HIV virus attack the CD4, T helper cell never undergo the activation. It never ever release the cytokines. If there is a no release of the cytokines, how does antibody will be produced? There will be a no production of the antibody. Entire immune system is getting collapsed completely. Kindly don't get confused with that. Okay, now you understand very clearly what is epitope. You also understand very clearly what is paratope also. What I can say, epitope is the immunologically active part of the immunogen or antigen. Paratope is the binding site where the epitope of antigen will come and attaches. Okay, so friends, before going to the different type of antibodies, their structures, their functions, their linkages, we have to compulsory understand about the receptor, and the receptors are exclusively important for the part C section. More than 2 times we got a direct question came from this particular receptor. Okay, every one of you listen my statements very carefully without confusion. Okay, listen my words. In our body there are different type of antigens or different type of immunogens are getting released, and most of the people will think that that every kind of a antigen or every kind of a immunogen can recognized only by this particular receptor or a single receptor only, which is absolutely wrong statement. Everyone keep it in your mind. In our body, our immune system contain different type of receptor, different type of receptor. It depending on the what kind of immunogen or what different type of epitope entered in our body, different types of receptors comes into the picture. So that's why we can call all the receptors as a pattern recognition receptor. What do you call, friends? A pattern recognition receptor. Yes or no? See, you take an example of bacteria. What certain bacteria will do? Certain bacteria will release their cell wall components. You know it very well. Correct, na? And certain bacteria release their flagella. We know that. Certain bacteria release their DNA. Certain bacteria release their capsule. If a virus enters in our body, virus releases RNA as their antigenic substance. So what is the point here? In our body, different types of antigens are entering. So now this different type of antigens can be recognized by the different type of receptor, or specific receptors can recognize the specific type of antigens that are entering in our body. It is not like only one receptor is going to recognize all the antigens that are entering in the body. It never happens like that. Suppose an example I am giving you. A gram-negative bacteria enter in our body. Listen to me, friends. It has released lipopolysaccharide as their antigenic substance. So a specific receptor comes into the picture which can recognize only lipopolysaccharide. A gram-positive bacteria enter in our body. Listen, you have to remember this. It has released lipotechoic acid as their antigen. So the point is very clear. Only the lipotechoic acid specific receptor only associated with it. Suppose example, the yeast entered in our body as an antigenic substance. Yeast released its cell wall components named as mannans. Or certain bacteria released glycolipids. Or certain bacteria released their bacterial DNA sequences. We can call CpG sequences as their antigen. So it doesn't matter whatever the type of antigens released by the bacteria or any other pathogenic organism, our body start responding specifically to that particular antigen only. Randomly only one receptor never ever go and recognize all the type of antigenic substances released by the pathogenic organism. For every antigenic substances released by the pathogenic organism, one specific receptor is there. Now listen, where these particular receptors are present, that is the point here. Don't get confused, you have to remember a lot of information. Now all these receptors, this I can say friends, 70% of the receptor, they are freely moving in the bloodstream. Whenever they find that specific antigenic substance, then they will go and recognize. See, all the students listen, 70% of them are freely moving in the blood circulation. Remaining 30% is what, friends? Remaining 30% will be membrane bounded, which means that they will be associated with a particular membrane only. Kindly don't get confused with that particular point. They are present. See, 70% of the receptors are present in the bloodstream as well as the tissue fluid. Remember, friends. And what are the best examples of the receptors which are freely moving in the bloodstream? Everyone pay attention. Mannose binding lectin receptor, C-reactive protein receptor, lipopolysaccharide binding protein receptor. These three are major freely circulating receptor in the bloodstream and in the tissue fluid. And the most important one which you people has to remember for the exam point of view, that is the membrane bound receptor. The two important membrane bound receptors are scavenger receptor, then followed by, friends, toll-like receptor. Among all, the most important one is the toll-like receptor, which was asked two times in the exam, especially in the 4-marker question only. Anyway, the name itself very clearly indicating, right? Here, these mannose binding lectin, C-reactive protein, lipopolysaccharide, you can see here, they are very specific receptor. Mannose binding lectin receptor can associated to the antigen which is of mannose only. Right? Same way, C-reactive protein is also very specific for specific antigen. So same way, this membrane bound receptors which are present on the membrane of the macrophages and other cells, they are also extremely specific. Anyway, friends, you no need to remember the functions at all, because all the functions are very similar. What you people has to remember is what kind of receptor is associated with which specific antigenic substance. That is what I want you people to remember for the exam, and that is only you will be expecting in the exam also. Every student pay attention. TLR2. TLR2, we will be calling it as, friends, generalized receptor. Okay. So friends, what do you mean by the word generalized receptor? I am using a word generalized receptor because many kinds of antigenic substances or many kind of immunogenic substances can be recognized by TLR2. What I mean to say, they are not extremely specific for only one specific antigen. So they will be specific for multiple. An example, a gram-positive bacteria entered in the body and it has released its cell wall components. Even lipopolysaccharide also can be recognized by the TLR2. Even whenever yeast entered in the body, whenever yeast released its cell wall component named as zymosan, that can also be recognized by this particular TLR2. Kindly do not get confused with this particular point. So that is the reason I will be calling TLR2 is the generalized receptor. Friends, requesting all of you remember this. Now you look into the TLR3. What the TLR3 is basically releasing? It is releasing the double-stranded RNA. Always remember this particular point. Whereas friends, TLR4 is basically recognizing what, friends? See, you have to keep it in your mind. TLR4 is specifically recognizing lipopolysaccharide only. It's a very specific receptor for the lipopolysaccharide. Kindly do not get confused with this particular point. And friends, what about the TLR5? TLR5 is very specific for the flagellin only. Whether a gram-positive bacteria released the flagellin or the gram-negative bacteria released the flagellin, doesn't matter. If a flagellin entered in our body, immediately TLR5 receptors will specifically responding towards that. And friends, if a particular bacteria released CpG as their antigenic substance, what is this CpG? Unmethylated cytosine and guanine sequence present in the bacterial DNA. That is what exactly we can call the CpG sequence. So that time TLR9 will be released. I hope last time also I have solved a question. Once again, the chapter is over, I'll solve it. Direct questions were asked from this particular receptor. Which kind of toll-like receptor can be recognized by the what kind of antigenic substance? A very direct and straightforward question which you have to remember. See, keep it in your mind. TLR9 CpG, TLR5 flagellin, TLR4 LPS, TLR3 double-stranded RNA, TLR2, I told you right, it's a kind of generalized receptor. All the students listen. This is the macrophage, correct na? This is the macrophage. On the membrane of the macrophage only what is present? TLR is present, correct na? Suppose example, this TLR recognized, see example, TLR4 which recognized specifically lipopolysaccharide. Now what is the function? What's happening over there? The moment TLR4 recognized the lipopolysaccharide, it immediately activates this macrophage. Even macrophage also, dendritic cell also. Immediately what is going to happen? The macrophages and the dendritic cells are going to release several cytokine molecules. It is going to induce several cytokine molecules. Now that cytokine molecules only will activate the further immune cells. Ultimately they will be involved in the production of the antibodies. So everywhere the function is common. Activating the macrophages and dendritic cell induce the secretion of the several cytokine molecule. Hence you people no need to remember the function. What you have to remember for the exam is name of the receptor and what kind of a specific antigen that it is recognizing. That is the only thing you have to remember. Okay. Now whatever we understand is the receptors, and we understand what is immunogen and what is antigen. We understand the differences between the immunogen and antigen. We understand two different type of immunogens, that is adjuvant as well as the hapten. Now friends, let's try to understand what is antibody, what are the functions of antibody, how we can cleave the antibody enzymatically. And I want every one of you remember one more important statement is that friends, these antibodies will be mostly important for the part B section also friends, as well as the part C section also. So please kindly do not get confused with this particular point. Okay, let's pay attention. Antibody. The first, how can you define the word antibody? It's a glycoprotein. So that's what you have to keep it in your mind. The antibody is not purely proteinaceous in nature. The antibody mostly protein, but it is also added with this sugar molecule. So it contains several sugar linkages. That's why we can call it glycoprotein. We know, right? We can also call it as them immunoglobulins. And who is going to secrete this particular antibodies? We know very well the B cells can be differentiated into plasma cell and memory cell, and this plasma cell secrete the secretory antibody, whereas memory cell can secrete what, friends? Membrane bound antibodies. Please kindly don't get confused with this particular statements. You will understand. Memory cell can secrete membrane bound antibody, plasma cell will secrete secretory antibody. Always keep it in your mind, the secretory antibody having short lifespan, because they are not associated with a membrane, so they are free, so they cannot survive for the longer time if they are not encountered with an antigen. But membrane bound antibody, they are associated with a membrane, so friends, they are having a long lifespan. Kindly don't get confused with that particular point. And we have clearly discussed that every antibody having a specific binding site. What do you call it, that particular specific binding site only, friends? We will be calling it as the paratope. To that particular paratope only, who will come and attach us, friends? Undoubtedly, epitope of the antigen will come and attach us. Such a simplified point it is. Now, friends, if you look into the specific structure of an antibody, every student has to listen. We can call antibody as the, everyone listen, you can call an antibody is the dimer of dimer. Dimer of dimer, or you can call it as what, friends? Tetramer. Why do I call it as the dimer of dimer? Because on one side two chains are there, on another side two chains are there. So this can act like one dimer and this can act like a one dimer. That is the reason we will be calling it as the dimer of the dimer. And you know, you can see the picture very clearly. On one side light chain and heavy chain, on another side also light chain and heavy chain. So what is the conclusion we can tell? We can tell a conclusion that an antibody consists of two identical light chains and two identical heavy chains. Always keep it in your mind, I am calling it as the heavy chain. What will be the reason? Because of their molecular weight, it's nearly 50,000 Dalton, and if you look into the light chains, they are just 25,000 Dalton. And friends, you all people can see the picture very clearly. This is the heavy chain, this is the light chain. How does the, listen friends, this is important one, don't get confused here. You can see the picture very clearly. The light chain and heavy chain connected with each other by what? The light and heavy chains are connected with each other by disulfide linkages. You can see that picture. Similarly, in this particular dimer also, the light and heavy chains are connected with the disulfide linkages. That also you can see. At the same time, friends, one dimer, this is called one dimer, right? And this is called another dimer. So friends, the two dimers can be connected with each other also with the help of the disulfide linkages only. Kindly do not get confused with this particular point. But what I would like to conclude a statement. I would like to conclude a statement that this is a very important one. When you look at the antibody, listen to me carefully. When you look at the antibody, if I ask you, friends, tell me which bond you can see that is connecting the one chain of the antibody to the another antibody, undoubtedly you all people can give me the answer that disulfide linkages. There is no doubt about it. But what I want you to remember, not only the disulfide linkages, there are other there are other non-covalent linkages also present which we can't see actually, but there are many other non-covalent linkages are present, such as salt linkages, hydrogen bonds, as well as the hydrophobic bonds. They are also involved to form a heterodimer. So they can ask you a question in the part B. What is the question they can ask? All of the following bonds are involved in joining the light and heavy chain except. So you people should not think that there is only presence of the disulfide linkages. There is a no other linkages are present, which is absolutely wrong statement. But what I would like to highlight is when compared to the all other linkages, friends, disulfide linkages are more in a number. Always remember that. Salt linkages, hydrogen bonds and hydrophobic bonds are present. But they are limited to very less in number they are actually. Please remember that, friends. Don't get confused with that particular point. Okay. Now every one of you listen my statements very carefully. We discussed that it is having 4 polypeptide chains. There is no doubt about it. Now listen to me. It consists of 2 identical light chains, which is kappa and lambda. What I mean to say, the 5 antibodies, everyone listen. The 5 antibodies consist of same kind of a light chain. There will be no differences in the light chain. Everyone keep it in your mind. There is a presence of 2 identical heavy chains. So what I mean to say, one antibody can be differ from the another antibody exclusively based on the heavy chain only. Kindly don't get confused with this particular point. We will explain what are that particular heavy chain. And we discussed that disulfide linkages are involved at the same time. Non-covalent like salt linkage, hydrogen bond and hydrophobic bonds are involved. And friends, I want you people to look into the picture. This is a heavy chain, correct na? At the same time, this is a heavy chain. See friends, this heavy chains, they contain a specialized area. You can see the picture. What do you call it, this particular specialized area? We can call it Hinge region. And what I want you to remember for the exam point of view is, what does that Hinge region is made up of? Please remember that the Hinge region is rich of an amino acid proline. Now you can ask me, sir, what is the major function of this particular Hinge region? Every student remember, they are extremely responsible for flexibility of the antibody. Flexibility of the antibody can be exclusively mediated by the Hinge region, and that Hinge region is exclusively made up of, friends, the amino acid proline. Please kindly don't get confused with this. Now whatever we are studying in this particular slide is the most important. Every student pay attention. It's a very important information. Pay attention. Most of the differences among the antibodies fall within the areas of the V regions. We can call the CDR. So you can get a direct in a given, listen friends, in a given 4 marker question, you will get one of the very direct statement. What is that statement? You will get one antibody can be differ from the another antibody majorly in their V regions called as CDRs, which is absolutely true statements. One antibody majorly differ with the another antibody in their V regions, which are having CDRs. Very important, friends. See friends, every student focus. This is the constant region, right? Constant light chain, constant heavy chain, constant light chain, constant heavy chain. Now what I want you to remember for the exam, friends, in the listen to me, you have to know very depth of the concept here. to the constant light chain and to the constant heavy chain, antibody never ever comes and attaches. Never, sorry, antigen never comes and attaches. Antigen will always comes and associated with the variable region only. That's why we have very clearly written here, antigen binding site is present in the variable regions only. You can see, right? Variable light region, variable heavy region. So all the students has to listen. Look into this line. On both light and heavy chains that constitute antigen binding site. My friends, antigen binding site or CDRs can be composed by the variable light chain and heavy chain. There is no involvement of the constant light chain and constant heavy chain. Now you can ask me a wonderful question. So then what is the function of a constant light chain and constant heavy chain if the antigen is only associated with the variable region? That's a valid question. Now the answer is here, the constant light chain and the constant heavy chain, they are responsible for the biological activity. What do you mean by that? I mean to say, now the antibody has to neutralize the antigen, yes or no? Antibody has to neutralize the antigen. So it has to show particular action. Now point is very clear, that biological activity can be mediated by the constant region, that is the constant light chain and constant heavy chain only, friends. Please kindly don't get confused with that. Whereas variable region only responsible for recognizing the antigen very specifically. The reason, variable light chain and variable heavy chain together, they make the CDR regions. Now all the students look into this picture very carefully. All of you pay attention, friends. This is really important concept. Now the question is here, how many CDRs are present? This is the CDR region, right? Every student has to look into this diagram very carefully. Okay, if you look into this diagram very carefully, in the middle there is an apple and two hands are there. Now you try to understand how many fingers are holding the antibody. Everyone pay attention. How many fingers are holding the antibody? Out of out of two hands, only 6 fingers, 3 fingers from one hand and 3 fingers from another hand holding the apple. Remaining any other part of the hand is not associated with this particular apple. So just to replace this finger, replace the hand with the antibody and replace the fingers with CDRs and replace the apple with the antigen. Now what is the conclusion? You can understand. Only 6 CDR region, 1, 2, 3, 4, 5, 6. 6 CDR regions only associated with this particular antigenic epitope. You can see the picture, CDR1, CDR2, CDR3. They are all present in the heavy chain region. Same way, in the variable light region also, CDR3, CDR1, CDR2. So friends, 3 CDR regions of heavy chain and 3 CDR regions of the light chain of the V variable associated with the antigen. This is only one side. So what I mean to say? I mean to say 6 CDR regions on one side, 6 CDR regions on another side. So what is the final statement I would like to conclude? The final statement I would like to conclude, one antibody, please keep it in your mind, one antibody having 12 CDRs. Never forget with that particular point. If you take an example of IgA, we're gonna read in the upcoming slides. We know, right friends, we can call the antibody IgA is the dimeric in nature. Dimer. See, whatever I have shown you is one monomer. One monomer having 12 CDR regions. Then the dimer having 2. 12 into 2, 24 CDRs are present in the IgA. Friends, if you take antibody like ICM, we're gonna read it, which is pentameric in nature. So one monomer having 12 CDR regions. So 12 into 5, 60 CDR regions are present in the ICM antibody. You don't need to remember for multiple units. You always remember, friends, for one unit. In one unit, friends, there are presence of 12 CDR regions are there. Kindly do not get confused with this particular point at all. Okay, all the students pay attention, friends. Look into the picture very carefully. Look into this. I am talking about very specifically CDR region. I am talking only about the CDR region. Constant light chain and variable light chain. Now you only tell me, do you think in the constant light region, that is CL, do you think I am talking about this and this? Now you only tell me, in the constant light chain, do you think is there any presence of the CDRs? No, not at all. So this is the 2D picture of a constant light domain, and this is the 3D picture of a constant light domain. Friends, I have told you in the biochemistry also many number of times. Always a beta, see, this is the secondary structure. You know, right? Beta pleated sheets are always buried where? Inside. And alpha helical structures are always exposed outer direction. You people can see it, right? Whatever this white color strands you are seeing, they are all alpha helical. And you can see arrows, right? What is that arrows? They are only, friends, beta pleated structures. Kindly do not get confused with this particular point. Now, friends, listen my statements very carefully. Here you can see the black color, right? What is this black color one, friends? That is the one which we generally calling it as the disulfide linkages. Very simple point it is. Now, but what is the statement I want you to understand? Here there is a no CDR, correct? Now you can take an example of 3D structure also. Friends, focus. All the beta pleated sheets are buried inside, and white color whichever you are seeing which are facing outside, they are what, friends? They are basically alpha helical structure. But now you look into the picture of variable light region. This is the 2D structure and this is the 3D structure of the variable light region. Look into the picture carefully. Alpha helix are there. Friends, beta pleated structures are also there. The black color one, the disulfide linkage also there. And friends, the most important and the significant point I want you to remember, there is a presence of the CDR regions also. You can see that very clearly, correct now? There is a presence of CDRs also. Now you can look into the tertiary structure little more elaboratively, friends. All the CDRs, see here my friends, look into this. All the CDRs come to one particular area, correct na? And this particular CDRs only acting as the binding site for the antigen. And what I want you to ultimately remember for the exam is the CDR regions are not associated with the constant region. They will be only associated with the variable region. Please remember that particular point without fail. Now, I have told you that there are 5 antibodies are there. And there is no doubt about it. Every antibody having the same kind of a light chain. Kappa or lambda. There is no difference at all. Then friends, it is very important. You people has to remember compulsory. You no need to remember the molecular formula, don't worry at all. What I want you to remember is especially their heavy chains compulsory. So one antibody can be differentiated with the another antibody with the help of a heavy chain only. Kindly do not get confused with this particular point. Friends, please don't get confused. IgG having a heavy chain gamma. I want you to remember it compulsory. IgM mu, IgA alpha, IgE epsilon, IgD is the delta. Please don't confuse and don't miss this. You have to remember this compulsory. Okay. And friends, once again the question which is asked many times in the exam, that is the chemical and mechanical digestion of this particular antibody. Always remember this. Please keep it in your mind. Different kinds of agents are going to show different mechanism of actions. What I mean to say? I mean to say different enzymes, different agents are going to cleave the antibody in a different ways. It's your responsibility to remember which enzyme and which agent is cleaving what part of the antibody. Very specifically you have to remember for exam. So you can see the picture. This is an antibody, correct now? This is what we can call, friends, FAB portion. We all people know it very well. And the most important portion, the tail part of the antibody, we are calling it as the FC portion. What is FC? Fractional crystallizable. What, friends? Fractional crystallizable. You people always remember as FC only. Okay, just remember as that. Now, when I treat an antibody with the first an enzyme called papain, what the papain has done, you know, friends, papain did not show any kind of a impact. Papain did not show any kind of a action on the FC fragments of the antibody. It left the FC fragments undigested. Once again, listen my statement. The papain left the FC fragments undigested. Keep that point in your mind. Whereas, friends, this particular papain completely cleaved two FAB portions separately. You can see that picture. This is one FAB portion, this is one FAB portion, and this is a FC portion. So what I can tell you? I can tell you that papain cleave the antibody into three fragments. Two FAB fragments and one FC fragments. And what is the point you have to remember? FC fragments are not at all digested completely, it just got cleaved, that's all. But friends, if you look into the pepsin, what is the speciality of the pepsin? It is just, it is not at all cleaving. What, friends? Pepsin is not at all cleaving any FAB portion. FAB portions are as it is, but what is the point? The pepsin, listen friends, listen. The pepsin not only cleaving the FC fragments. What I want you to remember, pepsin completely digesting the FC fragments. Pepsin completely digesting the FC fragments. Kindly do not get confused with this particular point at all. FC is completely digesting, pepsin is completely digesting the FC. You can't see the FC fragment. All the fractional crystallizable fragments got completely digested by the pepsin. This is the only one. I want you to keep it in your mind. Pepsin is the only enzyme which will do the complete digestion of FC. But if you look into the powerful and universal reducing agent which we commonly use in our laboratories, mercaptoethanol and friends DTT, dithiotritol. These two are the reducing agent. And friends, all the students remember common point. almost all the reducing agents, friends, they break the antibodies into 4 parts. They will separate into 2 light chains and 2 heavy chains by specifically acting on the disulfide linkages only. Kindly do not get confused with this particular point, friends. Light chains, heavy chains can be separated by a mercaptoethanol and dithiotritol, and pepsin cannot attack the FAB fragments. It completely digests the FC. Papain can cleave the antibody into 2 FAB fragments and 1 FC fragment. That's the simple concept of the enzymatic digestion. So friends, so far what we read what is immunogen, what is antigen, the differences. We read all the type of the receptors, and we read the structure of the antibody, and we also understand the enzymatic cleavage of the antibody. And now we have to majorly concentrate on the functions of the antibody. Okay, so we will discuss these functions of the antibody in the tomorrow class. Okay, and friends, as I told you, we have to start the developmental biology simultaneously. Okay, so we will do the 2 chapters simultaneously today itself. I will be starting the developmental biology, no doubt about it. So we will be having a class today at 4:30 p.m. Okay, I will share the link of this developmental biology class. I am requesting all of you to join the class. We will be starting today itself. Okay, I'll finish the developmental biology very faster. Okay, in 15 to 16 classes we will finish this. Okay friends, I'll see you in the afternoon class. Okay, please attend without fail. Okay friends, that's all for now. I hope the discussion is useful. If you have any kind of a doubts, you can ask. Hope it's clear for everyone. Okay, thank you all. I'll see you in the afternoon class. Okay friends, thank you all. Thank you all.